Well, hello, welcome back, or welcome to IBD Edinburgh 2022. This is our final session. This is our keynote, and I know many of you will be joining us specifically for this. We are now going to go into COVID and IBD. We really have the pleasure of not just having Nick Kennedy sat next to me as co-chair, but we have Jeff Barrett, Nick Powell, and Tariq Ahmad, three world-leading experts on this condition now to help us unravel where we are at. So, without further ado, I am going to introduce Jeff Barrett. Jeff is coming to us live from Helsinki. Jeff is very well known to us as a bioinformatician, a computational geneticist who has worked in the field of IBD genetics for well over a decade. Jeff has been spending time working to advance the true translation of IBD genetics and more generally complex disease genetics. But over the last 2 years, Jeff has had an incredibly important role, a really pivotal role, right at the heart of the COVID pandemic, been instrumental in understanding what is going on with the genetic profiling or the evolution, the sequencing of the SARS-CoV-2 virus that is, of course, responsible for all the misery over the last couple of years. So what we're going to do is Jeff is going to give us an update on COVID variants and sequencing, and then we're going to go to Tariq and Nick for the clinical aspects, and then we're going to have a cracking Q&A. Submit your questions through the panel as before. We will come to all of those. We've got plenty of time for a great Q&A. Jeff, great to see you, my friend. Please kick us off.
Thanks, Charlie. Very happy to be here. So yes, as Charlie mentioned, my name is Jeff Barrett, and for the last 2 years, I've been at the Wellcome Sanger Institute in Cambridge, leading the Sanger's COVID sequencing initiative, COVID genomics initiative. And what I'm going to tell you about is how we have developed a system to basically track with unprecedented precision how this virus has evolved and moved around the country using a very high throughput genome sequencing pipeline. So all of us have become accustomed to looking at graphs like this and seeing the pattern of the pandemic over time. This is the UK case graph as of this morning, and it has a sort of recognizable shape. Of course, it's worth remembering that if we look at the entire thing, the very beginning of the pandemic here and basically everywhere in the world is missing a whole bunch of cases because we didn't have testing infrastructure at that point. So I've kind of made up a line to give you some guess of what the true shape of the pandemic has looked like. And actually, it's kind of amazing having talked about this, that every time I think I've sort of got to the point where we've finished with the story, that some massive change happens, of course, most recently with the arrival of the Omicron variant, which has rescaled y-axes everywhere. So what the Sanger did as part of something called COG UK, the Coronavirus Genomics UK Consortium, starting in the summer of 2020, was to put together an approach to sequence the genome of the coronavirus from positive PCR tests. And Sanger's particular approach was, rather than, say, work with a particular hospital and track outbreaks in a particular place, for example, which some of our partners in COG UK did, was to develop a relationship with the so-called lighthouse labs, which are the large centralized testing facilities around the UK that scaled up in 2020 and are now processing millions of tests every single day. And we basically set up a system where these chilled vans bring hundreds of thousands of the waste material, basically from the PCR tests, to the Sanger Institute in Hingston, just outside of Cambridge. We, early in the pandemic, put a few giant trailer-sized freezers on one of the parking lots, and they're still there. They're absolutely full of these plastic boxes full of samples. We first with volunteers, and over time kind of built a professional setup to every morning be unpacking these massive boxes, pulling out 96 well plates full of waste material from the PCR runs, putting them onto a set of robotic pipelines that, again, we've developed over time that pick out every day a representative random geographic sample from around the country, plus a few other special cases, things like trying to sequence as many travelers as possible during times in the pandemic when that's an important feature, and then putting them all onto high throughput Illumina sequencing machines. And when we started in earnest, so we did various things sort of in early 2020, and we really got going in September of 2020. And then we were doing probably a couple of thousand genomes a week. It was the biggest sort of shop in the world in terms of genome sequencing, but it has absolutely grown massively since then, to the point where now the initiative in just one place is sequencing 64,000 genomes every week, week after week. If we look at what we learned over time, we knew that tracking how the virus was evolving and seeing when we could detect outbreaks and whether they were driven by the same lineage of the virus would be valuable, but we really didn't know exactly what was going to happen. And early on in late 2020, we were kind of pitching to the scientific community, to the government, that this very detailed surveillance was going to be valuable, and we were learning how it would be so. So if we cast our minds back to late 2020, the kind of mini lockdown in November was ending. People were going back about their business. Cases were on the way down after a very quiet summer. And what happened, of course, was cases blew up in around Christmas time. And if you look at, if you remember, this seems like another lifetime, but the top row are maps of England by the tier system in late 2020, where red is kind of eventually we all landed in tier 4 in early 21. But you can see as the sort of restrictions were put in geographically over time, what we discovered, because we weren't just looking at case numbers, but we were able to see the genome sequences in a proportion of these to separate them. Along the bottom were the kind of maps of the patterns, the growth patterns of the ancestral strains of the virus, and in the middle was what was this at the time anonymous variant of the virus called B117, which had a whole bunch of mutations we hadn't seen before, later now known as the alpha variant. And blue here means the R epidemic number is less than 1, so the epidemic is shrinking, and red means it's growing. And you can see during the light lockdown, all the sort of original COVID was going away. In red, things were growing. This is alpha, which was sort of under the radar growing, especially in the southeast and Kent. What we can see is that it was absolutely taking off through December when the geographic restrictions just weren't sufficient. And then, of course, famously, the Prime Minister cancelled Christmas, brought in nationwide lockdown. And you can see by the time we get to the right-hand side of this, everything is blue. The kind of the older versions of the virus are more blue than alpha was. But it was good news because it meant that this was just as the vaccines were being rolled out, but the strict lockdown was enough, even with this new variant, to bring things back under control. And that was our very first taste of seeing that the virus that erupted in the world in late 2019 and spread throughout 2020 was going to change. In this case, it developed a much higher amount of inherent transmissibility, and we had to adapt to respond to it. This, of course, wasn't the last time that things changed. So then in late 20 and early 21, we had the lockdown, we had the successful vaccine rollout, and that brought things under control to the summer of last year. And actually, I think it's very likely that if the virus had stopped evolving, the pandemic really would have ended last year. And because the alpha variant was so well neutralized by the first current generation of vaccines that it essentially went extinct in England and indeed in almost any place where vaccine coverage was good. However, around this time, the world watched extremely gruesome scenes in India as they had a massive wave of COVID in Maharashtra, where Mumbai is starting earlier, and then even more acutely in Delhi. And the question was, because at the time, the surveillance sequencing in India was okay, but not brilliant, what was driving this? Was it a new variant or not? And there was a long debate in the Western world. And as it happened, the UK ended up being, and this was, I remember just the trying to get actionable intelligence. And there was a moment when the best information you could get was by watching a YouTube stream of some Indian scientists who were sharing their sequences in real time. And there has really been a sort of lesson of this, of trying to build more sustainable ways to share this data. But what ended up happening was that actually the UK surveillance, because of the heavy travel links between India and the UK, and of course there were political consequences around decisions in this regard, but a large number of introductions of what's now known as the Delta variant arrived in the UK in midsummer of 2021. And the massive amount of sequencing we were doing allowed the UK to really be kind of beacon to say, this is a new variant. It has fundamentally different properties even than alpha. It is far more transmissible, and therefore we need to be looking at it very carefully. And finally, again, as I said, I have to keep updating these slides. I kind of thought we'd be in a long, holding position with Delta, but there was then another new variant, in this case, first detected in Southern Africa. In South Africa, they actually have excellent genomic surveillance, and so they made very rapid progress in announcing this new variant, which had an even more heavily mutated spike gene, the protein that's used to bind to human cells. And before it even spread very widely, I think the world knew something was going to be up. And this was actually the Slack channel from the first observed Omicron variant in England. We were sort of, in this case, it wasn't that it was trickling in and we were trying to figure it out. We knew this was coming and we're watching for it. And this was some late evening, just a few days after South Africa had announced it to the world, that we detected the first one, with a turnaround time now of the swab up someone's nose to the sequence being analyzed of just a couple of days. And of course, Omicron is spreading extremely fast in the UK and indeed around the world. And I've kind of only told you the highlights of this, but the system that we build really means that in England, at least, we can look at this curve not just as coronavirus, but we know at every point in the timing of the pandemic exactly what was happening. So, in the fall were the sort of older versions of the virus that now seem quaintly not contagious. This purple peak was the alpha variant that we first realized the virus was evolving in meaningful ways. This long green plateau was the time of Delta. And then this almost unbelievably sharp peak of yellow is now the Omicron spike that we're living through at the moment. And just to emphasize, there's something like 1.4 million viral genomes under this picture, and the whole world has publicly shared 7 million. So Sanger has sequenced 1 in 5 coronavirus genomes in the entire world. Okay, what I want to finish with, just mention briefly, there's a website, covid19.sanger.ac.uk, if you want to explore these data in real time. They get updated every week. Just briefly before I hand over, because I think it's relevant to what Tariq and Nick will talk about and to the discussion, is some of the molecular phenotyping of the Omicron variant. This is an antigenic map. So these are previous variants, and each square is serum from a vaccinated individual colored by which vaccine they had. And basically, this is a kind of multidimensional projection of how well these different sera can neutralize these variants. And you can see we were kind of worried that the beta variant, and to a lesser extent Delta, were a bit far from the original Wuhan virus. But Omicron is just practically on a different page. And this is the fundamental change. Omicron has a massively reduced neutralization by existing immunity antibodies, either from vaccines or infection. And that's the thing we're confronting right now. The transmissibility is a kind of open question, but it is just absolutely racing through people who've been previously infected or who have been vaccinated. There's some interesting data that I could do a whole 2-hour seminar on all of the lab data that different labs have published. I just want to draw attention to this beautiful paper from Tom Peacock and Wendy Barclay's lab. If you compare Delta and Omicron, there's very briefly 2 ways that the virus can enter cells, one which requires cell fusion and the other which doesn't. And this drug Camostat inhibits the cell fusion pathway. And so you can see Delta grows fine when not inhibited, but it heavily relies on that pathway and does not grow if you knock it out. Omicron grows faster regardless, but is basically, it is affected, but much less so by this inhibitor. And that inhibitor is really about an ACE2, which is the primary receptor partner called TMPRSS2. And what's happening there is probably Omicron is a more generic pathogen that can infect not just lung cells, which have both ACE2 and TMPRSS2, but upper airway cells, which have a lot of ACE2 without TMPRSS2. And that change in the tissue area seems to be reflected in a lot of the possible differences in the severity and phenotype. That's early data. All of this means that if you look at vaccine data, the black dots are Delta, the gray dots are Omicron. The left-hand side is with 2 doses. The right-hand side is after boosting. This is from symptomatic infection. Omicron, as I said, just blasts through antibodies. If you've had 2 doses of just 2 doses of AZ a long time ago, you have almost no protection from symptomatic infection. It does thankfully get much higher when boosted. And even better news, the protection from hospitalization and severe disease is better preserved, and especially after boosting. And there is an inherent more mildness in Omicron, at least in adults, That may not be true in kids. When you put all this together, though, because we have such a huge number of cases, even though through the existing vaccine immunity and the mildness per person, you don't get as much severe disease as anyone who's currently working in the healthcare system can attest. There is a massive strain as we try to get through this current peak. And I think what hopefully will come up in the discussion is, especially for groups of patients who may have compromised immune systems, the balance, I think, right now and for a few more weeks or maybe a month is going to be quite fine. Okay, thanks very much. Now let me hand over to Tariq and Nick.
Thanks very much, Jeff. Excellent talk, and it's really wonderful to see these data being presented in such a clear way, charting out what's happened during the pandemic. So the last 2 years have seen all sorts of changes, and amongst many others, one of the changes we've seen has been a pivoting of some of those of us who are doing IBD research to looking at the impact of COVID-19 on our patients with IBD. And I'm delighted to introduce our next two speakers, Nick Powell from Imperial and Tariq Ahmad, my colleague from Exeter, who are going to talk through the work that's been done in this area, and particularly about the VIP study and the Clarity study, which have really led the world in terms of looking at the relationship between IBD drugs and COVID-19. So Nick Powell I'm going to introduce first, and then we're going to have a double act from the two of them over the next little while.
Great, thank you so much, Nick. So we're going to talk mostly over the next sort of 20 minutes or so about COVID-19 vaccination in the context of our IBD patients. And I should say my co-speaker today, Tariq Ahmad, and friend now actually over the last sort of 18 months or so, as well as many of the other members of the Exeter team. And actually, Tim Rain hit on a really important point that's happened during this pandemic. And that's how we've done these two big studies we're going to talk about. That's going to be the key focus of this, but how it's brought together a really cohesive feature amongst IBD researchers across the UK. And that's been a real privilege to play a part in that. So that's a really important thing to stress. So these are our disclosures. This is what the agenda is going to be. So I'm going to just briefly touch on IBD and COVID-19 and what we know about that so far, but really then we're going to drill into vaccination. What do we know about more generally about vaccination in IBD? And then we're going to serve up the main course, which is really reporting back. And this is the first time these data have been properly aired. Two important prospective studies, Clarity IBD and VIP. So we're showing you new data. Both of these papers, actually, I'm glad to say, have recently been accepted for publication, so you'll get your hands on them soon as well. But really that main course is going to be looking at the immunogenicity of vaccines. How well do they elicit an immune response in our IBD patients, and particularly in the context of our concern here, and that's that immunosuppression might just blunt that response. So that's a really key part of the talk. What about the durability of those responses? That's another key or really important aspect about how effective these vaccines are likely to be. And of course, the ultimate proof of pudding is, do they actually prevent infection or reinfection? So that's going to be the main bit. And then Tariq will also finish on, again, what I think some really exciting data, and that's the impact of the third dose or booster doses in the context of our IBD immunosuppressed patients. So just briefly, to just set the table for COVID-19 in IBD, what do we know about that so far? Well, I think this is probably one of the better sources of data that we have so far. So this is an American database, Secure IBD, that reports IBD patients who have had COVID and looks at their outcomes. And what have we learned from this? Well, it's a decent sized study, so over 6,000 patients. It's North America heavy. A good size of those patients are from the States. But if you look at what happens to patients when they get COVID-19, if you have IBD, reassuringly, I think they look very much like what happens to people in the general population. So about 15% of them end up hospitalized. About 3% land themselves in intensive care, 2% require ventilation, and the overall case mortality is about 2%. And this is mostly in the pre-vaccine era, I should say. And you remember in the UK, in the pre-vaccine era, the case fatality rate was also about 2%. So whilst on one hand that's reassuring, this is also a global pandemic that's killing lots of people still. So that still should be a big important thing for us to think about. And what drives bad outcomes in IBD patients? So again, these are data from Secure IBD. Unsurprisingly, increasing age is a really important driver, as we see in the general population. Males tend to do a little bit worse too, again, as we've seen in the general population. And no surprise, comorbidity is a key driver of poor outcomes in IBD patients. And then this is an interesting one, I think, for the IBD community. Active disease is also associated with poor outcomes. So twice the rate of dying, requiring ITU or ventilation if you've got moderate to severe disease. So what's our escape strategy to this? So you could be mistaken to think that it's drinking wine and cheese, but it is not. The escape strategy to this is vaccination. And of course, we've also embraced that. And I heard today that JVT has actually resigned from his post. That's him vaccinating there. And it's really key that if we are going to escape this pandemic effectively, then good vaccination strategy, knowing what vaccination does in our patients is going to be pretty important for IBD patients. These are data we've known for a long time now about how immunosuppression potentially impacts how effective our vaccines are, or at least how good they are at eliciting effective antibody responses. These are data on the left-hand side here from California, looking at response to pneumococcal vaccination. And you see a nice good response in healthy controls, a good response in IBD patients if you're taking mesalazine, but those patients who are taking both immunomodulator and anti-TNF having a plummeted or a reduced response, which is of course a concern. And then on the right-hand side, we tried to put together here a heat map that looks at different vaccines that are given in IBD patients according to their therapies, looking at pneumococcal vaccine, hepatitis virus vaccines, as well as a host of influenza strains. And again, you can see in that heat map, it's looking pretty hot in the anti-TNF group, and also a little bit pink for some of those immunomodulator groups in some of those studies. So you can see that this is of concern for our patients in the context of a really key escape strategy. That hands over nicely to my colleague Tariq, who's going to talk to you about Clarity IBD, which is a really key study, I think, that helps address exactly some of these questions we've been discussing.
Thank you, Charlie. I'm sorry I couldn't be with you in London today. So I'm going to talk about Clarity IBD, which is a study that many of you will have been involved in. And the overarching aim was to investigate the impact of immunomodulator and biologic therapy on SARS-CoV-2 infection and immunity in our patients with inflammatory bowel disease. And this was an NIHR supported study, as it was designated as an urgent public health study, which made it possible for us to deliver this work on time. So there were a number of study objectives, and the list has grown or evolved during the course of the pandemic. But today, I'm just going to focus on the impact of immunomodulators and biologics on the outcomes in bold here. So anti-SARS-CoV-2 spike antibody levels and rates of seroconversion following 2 doses of vaccine, and later on in the presentation, 3 doses of vaccine. Importantly, the durability of antibody responses in patients receiving these medicines, and then the risk of breakthrough infections in our patients following vaccination. So this was initially set out as a 40-week study. In fact, we extended this for a further 24 weeks, and we recruited just under 7,000 people with IBD age 5 or over who were receiving infliximab or vedolizumab at 92 hospitals, shown here on the map on the left. And we chose infliximab for the reasons that Nick has just mentioned, and vedolizumab treated patients were the control group that we used. And we use these individuals principally because they were also coming into hospital and receiving infusions at the same frequency. So all of these patients were recruited in a 12-week window running up to Christmas 2020. Importantly, we were granted permission to handle personal data for these subjects in Exeter, and this allowed us to carry out e-consent, but also to communicate directly with patients, to push out questionnaires so that these could be completed, but also to update patients on their own results and the results of the study in general. We also used the NHS number to link to Public Health England, Scotland and Wales, so that we were able to access PCR testing results. And although it's been a bit more difficult, we hope to also link to the national vaccine data as well. So this is the schedule of study visits. Essentially, when patients attended every 8 weeks, typically every 8 weeks for an infusion, they were asked to complete a questionnaire and have a blood sample taken. And these were then sent to Exeter by Royal Mail. The questionnaire collected information regarding disease activity, isolating strategies that the patients were adopted, symptoms of COVID-19, and also information regarding depression and anxiety. And then we asked sites to complete further information at visits 1, at visit 6, and visit 9, the last visit. And most of our participants are now out to the end of the study, or shortly will be reaching it. So we used the Roche antibody assays. Initially, we used an antibody against the nuclear capsid part of the virus, and that tells us whether somebody has had past infection. Later on, Roche developed an anti-spike antibody assay, and we used that in parallel with the anti-N antibody. And this combination of tests allowed us to look at antibody responses after vaccination, but also to recognize when people who had been vaccinated had a subsequent infection. Importantly, we returned these results back to patients. So patients, we texted patients, they could log into the system and retrieve their results. And I think that was appreciated by many patients in this study. So what I'm going to do first of all is just show you the data in antibody responses following 2 doses of vaccines. And I don't expect you to read the data on this slide, but basically this is based on 1,024 patients treated with vedolizumab and 2,264 patients treated with infliximab who had serum samples available 2 to 10 weeks after the 2nd dose of vaccine. And there were some important differences in these patient groups that's worth mentioning. So vedolizumab treated patients, as you might expect, tended to be older. They were less likely to be taking a concomitant immunomodulator, and they had a greater number of comorbidities. So important differences between the 2 patient groups. So this first data slide shows anti-SARS-CoV-2 antibody concentrations after 1 and after 2 doses of the Pfizer vaccine. So first of all, we've got the antibody levels for infliximab treated patients in green. And on the y-axis is a logarithmic scale. We've got the antibody concentration. And you can see that after 2 doses of vaccine, there's a very significant increase in antibody concentrations, but they are approximately 10-fold higher in patients treated with vedolizumab. So that's the data for the Pfizer vaccine, and here are the data for the AstraZeneca vaccine. And we see this similar increase, although the absolute antibody levels are lower, as you might expect, perhaps with the AstraZeneca vaccine. We also looked at the other factors that were independently associated with antibody concentrations, and the numbers here are the fold changes of antibody concentrations associated with each of the factors on the left-hand side. And you can see here that infliximab clearly exerts the most marked effect in lowering antibody concentrations, but lower antibody levels are also seen in individuals taking a thiopurine, those taking methotrexate, steroids, people with Crohn's disease rather than ulcerative colitis, older patients, as you might expect from all the other studies that have been carried out, and interestingly, current smokers. In contrast, higher antibody levels are seen in people vaccinated with Pfizer and in people of non-white ethnicity. So there are the independent factors. And here are data relating to decay in antibody levels over time. So if we look at the top half of the figure, first of all, and these data are timed from the second dose of vaccine, this vertical bar. And as you can see, the peak antibody concentration after 2 doses of vaccine is lower in people treated with infliximab, shown here in green, compared to vedolizumab, as I've just shown you. And the decay is quicker in people treated with infliximab. So by the time we reach 24 weeks from the second dose of vaccine, the level here is approaching this somewhat arbitrary seroconversion level of 15 units per mil. In contrast, patients who are treated with vedolizumab, the antibody levels remain at a relatively high level out to the same time period. And we see a similar effect looking at this rolling geometric mean for patients vaccinated with the AstraZeneca vaccine. So does this really matter? Do the antibody levels actually have any clinical adverse clinical outcome? So here are data on breakthrough infections. Now this data was cut in September, so prior to Omicron, and obviously some months ago. This is, at this point, 5% of our study population had had a breakthrough infection more than 2 weeks from a second dose of vaccine. On the left here, we've got time to positive PCR test prior to a dose of vaccine. And you can see there's no difference between vedolizumab and infliximab treated individuals. However, after 2 doses of vaccine, you can see that there are more infections and the time to infection or breakthrough infection is shorter in infliximab treated patients. So these individuals do appear to be at greater risk of breakthrough infection. Now, reassuringly, at the time this data was cut, of the people with breakthrough infection, only 1% were hospitalized. So that was certainly reassuring for individuals. So you might be at a greater risk of acquiring the virus, but you're not getting sick with it. So if I summarize the first part here, after 2 doses of vaccine, infliximab treated patients have lower, less durable vaccine-induced antibody responses and an increased risk of breakthrough infections. We observe a higher peak antibody levels and lower rate of breakthrough infections with the Pfizer vaccine. I haven't shown you all of that data, suggesting that the Pfizer vaccine is most appropriate for infliximab treated patients rather than the AstraZeneca vaccine. Based on these data, after 2 doses, we believe that anti-TNF treated patients should receive, as they have done, of course, a 3rd primary dose of vaccine. And the Clarity data supports this, the JCVI recommended interval of 8 weeks after the 2nd dose. Now, although we haven't specifically looked at other anti-TNF drugs, we suspect that we will see similar effects for other anti-TNF medicines and, of course, across other indications. So at this point, I'm going to hand back to Nick Powell to consider what happens when you use other medicines to treat our patients with IBD.
Great, thanks, Tariq. So very clear that there's an iceberg ahead for infliximab-treated patients. What about our other drugs? That was the other question we were hoping to address with the VIP study. So VIP, again, was a prospective, multi-centre, UK-based study with a clear aim to just look at the impact of COVID-19 vaccine-induced immunogenicity in patients with different immunosuppressive treatments. So very straightforward study design. Again, a bit unlike Clarity, we wanted to have some differences here. We recruited a healthy population, so patients without inflammatory disease, patients without any immunosuppressive treatments, and then many of our key treatments that we use regularly in inflammatory bowel disease. So thiopurine monotherapy, infliximab monotherapy, infliximab thiopurine combination therapy, and again monotherapy with ustekinumab, vedolizumab, or tofacitinib. And again, you can't have a pragmatic design here, getting people's blood post-infusion, because they've all got different treatment regimens. So we fixed the serum sampling at a fixed time interval after the second dose of vaccine was administered. And the primary outcome in this case was, again, looking at the anti-S RBD antibody concentration, and really importantly, the primary predefined endpoint was in patients without prior infection. We know that's a key confounder here, but also in those patients stratified by treatment, we also adjusted for age and vaccine type, because we know these are really key influencers of the serological response. Again, it was pretty much a nationwide study in some sense, on a more limited sense than Clarity was, and it was slightly London heavy. You can see there's several participating sites. And also a big thanks to Edinburgh. When we were struggling for recruitment, Edinburgh were the last people to join and really quickly caught up. So big thank you to the team there. And of course to all the other teams that contributed to this. This was done at very fast pace. It was a pretty tricky study to deliver in some ways, and getting that fixed time point during a pandemic when people didn't want to come into hospital. This is what the study population looked like. So I think it was nearly 500 patients, and we've got some additional patients to analyse here, but about 120 healthy controls. And again, you can see the 60 or 70 patients in thiopurines, infliximab, combo therapy, usti, vedo, and tofacitinib. I will just draw your attention to the lower number of patients in that tofacitinib group, though. I won't spend ages on the demographics, because it won't surprise you that the demographic differences don't look that different to how you saw in Clarity, and Tariq's already described that in some detail. What I will mention, though, is that you'll notice that there was a difference in the type of vaccine that was administered to our control population in comparison to some of our treatment groups, with the majority of patients in the control arm getting the Pfizer vaccine, or an mRNA vaccine, which was heavily influenced by Pfizer, whereas our other treatment groups were either balanced or slightly tilted towards the AstraZeneca vaccine. And this was one of the key reasons we kind of anticipated this, and this was one of the reasons we had a sort of predefined vaccine-adjusted endpoint. So these are just a crude architecture of the data. So this is unadjusted for age and vaccine type, but just to give you a general feel of what those data look like. So do take them with a pinch of salt, but I don't think it's going to surprise anybody, having seen the last Clarity presentation, that you can see a pretty marked impact on infliximab and thiopurine infliximab combo therapy group. But at a glance, you'd be pretty reassured here if you're taking thiopurines, I imagine, and you'd perhaps cast your eye across to the right-hand side of the graph and suggest that maybe that tofacitinib group, even though it's a small number, does look like they could have some impact as well. So, but what about the primary data now, adjusting for age and adjusting for vaccine type to account for some of those differences we saw in our demographic group? So I'll just go through these here. So if you look at that top line, the top group are thiopurine treated patients. And if you're on that dotted line, that means the geometric mean ratio of your antibody concentration looks pretty much like it looks in a healthy control population. So that's good news for thiopurine treated patients. What about infliximab? Well, this is not going to surprise you, having seen Clarity, a really marked deficit in antibody production in infliximab-treated patients, almost a tenfold reduction, and a similar finding in the combination therapy group. The other thing you'll notice in all of these settings is if you had an mRNA vaccine, you had a better antibody response as well. And again, almost universally, you saw an impact of age. So again, we were correct to adjust for age. You can see that age did drive a poor antibody response. And what about the other drugs? So ustekinumab, again, you can see that that error bar, that 95% confidence interval, does just cross one, so that was not statistically different from healthy control serological responses. Likewise, vedolizumab actually looking very like healthy controls. But tofacitinib, you're seeing an antibody response about half that you would see in a healthy control population. Again, universally, you're seeing that increased serological response if you had an mRNA vaccine, and the role of age in some but not all of those groups. What about the impact of prior infection? Again, so that primary analysis excluded all the patients that had had a primary infection. But what did they look like when we compared them? So again, this is the anti-S antibody on the y-axis and the treatment groups along the y-axis with plus or negative infection. So there's no infection in the first group and then with infection in the next. And you can see across the board, really, if you'd had a prior infection, your response was better, supporting this notion that a third antigen exposure is sufficient to boost your response. And of course, that's what underpins the booster and third dose situation. So just to summarise the VIP findings, so clearly vaccine-induced anti-S antibodies are impaired in both anti-TNF and tofacitinib treated IBD patients, but perhaps no new wrinkles for thiopurines, vedolizumab, and ustekinumab. And I think it's also clear that if you've had a prior exposure to antigen, 3 hits of antigen, your response is pretty substantially improved. And again, across the board, really, no matter what treatment you're on, if you had an mRNA vaccine, your response was really pretty good. So that takes us to sort of third dose. So Tariq's now going to talk to us about what happened in the Clarity cohort in those patients who we've had a glimpse of now of those who've had a third or booster dose of vaccine. Tariq.
So the data that I'm going to show you in a second is hot off the press. It was put together by Nick Kennedy yesterday, having received it on Monday. So this is new. So this slide shows what's happened to our cohort of patients. We followed them up to this week. And this slide, first of all, shows the proportion of patients treated with infliximab at the top and vedolizumab underneath each week who have a positive PCR test. And you can see, first of all, that up to about 3% of infliximab treated patients in the last few weeks have had a positive PCR test within this cohort. The fewer patients treated with vedolizumab have had a positive PCR test. This data here also shows information about Omicron indirectly. So here we've looked at S gene target failure. Now, for people that don't know about this, most of the labs in the UK use the TACPath PCR assay, which has 3 gene targets. And one of these is knocked out by deletion that's carried by both Omicron and the Alpha variant. So that's why you can see it also earlier on in the pandemic. So these people with S gene knockouts are in blue. People without the S gene knockout are in red. So you can see that the vast majority of people in recent weeks for whom we have data on S gene knockout are positive for this and therefore have Omicron. So a massive increase in cases, majority of which are Omicron, as you might expect. So we've carried on looking at these patients, and now we're looking at antibody responses timed from the third primary dose. And what you can see here is that there is a very significant increase in the peak antibody concentration after 3 doses compared to the peak antibody concentration after 2 doses. So this is in people without prior infection on the top half of the graph. But it's clear that for Pfizer on the left here, that there is still a very large difference in antibody concentrations in infliximab treated patients compared to those treated with vedolizumab. What's interesting, after 3 doses of vaccine, the antibody levels achieved are very similar, regardless of whether your first 2 doses were with AstraZeneca or with the Pfizer vaccine. Obviously, for the 3rd primary dose, everybody got Pfizer. So it doesn't matter, it doesn't appear to matter that your first 2 doses were AstraZeneca compared to Pfizer. But what we can see from this very early cut of the data is that antibody levels are again starting to fall, particularly in the patients treated with infliximab. So again, does this matter? So here are some data on breakthrough infections. Again, this is an update of the figure that I showed earlier in the presentation. You can see that prior to vaccination, there is no difference in breakthrough infections, vedolizumab versus infliximab. Following 2 doses, there are more infections. You can see here breakthrough infections approaching 40% in infliximab-treated patients, perhaps half that number in vedolizumab-treated patients. But this is the sort of dramatic figure at the end here. After 3 doses, really the time to infection, PCR confirmed infection, and the number of infections is considerably greater in infliximab treated patients. So at the moment, we are trying to get some data on severity for these individuals. We're putting this together, but I think it's clear that the vast majority of these patients are not being hospitalized. So certainly for Omicron, this doesn't appear to matter too much. But of course, if we were to have more serious variants in the future, this may become a problem. So just to summarize the 3-dose data, following a 3rd primary dose with Pfizer vaccine, antibody concentrations increased 10 to 30-fold compared to peak antibody concentrations after a 2nd dose. Again, infliximab is associated with attenuated antibody responses. And breakthrough infections and reinfections, and I've not shown you the data on reinfections, but both breakthrough and reinfections are more common and occur earlier in infliximab-treated patients, irrespective of the initial vaccine type. So I think we conclude by saying that we suspect that more intensive immunization strategies may be required for patients treated with anti-TNF therapy. So Clarity is winding up. I'm grateful for everybody that's been involved in this study. Nick and I both want to thank, first of all, the 92 principal investigators and more than 400 research nurses that have collected data for this study. There are some individuals that have gone above and beyond. Claire Boucher, who has worked night and day being the study manager for both Clarity and VIP. Rachel Nice, who's done all the serology work. The team in Imperial, particularly James Alexander, who's led the VIP study, done a lot of work. My colleagues in Exeter, Nick and James, who have been absolutely pivotal to delivering this work. It wouldn't have happened without them. And Charlie Lee's there in the middle for being our PR expert during the course of the study, delivering results across the world and to all the patients. So thanks very much.
Well, thank you, Tariq. Thank you, Nick. Thank you, Jeff. There are questions coming in, and I know some of our co-faculty want to come in from the floor. I just briefly want to come to each of you in turn with a brief comment on what next. Tariq talked about until the next variant, so that seems an obvious segue to Jeff to say, oh, please no. I'm sure lots of people would be thinking, please no. But let's just have a thought from Jeff and then Nick and then Tariq. What next? And then we'll take some further questions. Jeff.
Well, I've tried to avoid making predictions in this pandemic because it's so easy to be wrong. I certainly hope that we aren't going to be faced with exactly this story again in a very short time. And I think there's some reason to be hopeful. The two in mind are the world is getting a massive dose of immunity from this Omicron wave. And it comes with problems, of course, but it also means that after, and we're starting to see in multiple countries, including the UK, the case peak at least seems to have happened, that it goes through very hot and very fast, and then what it leaves is a lot of immunity. And there is some early data, at least, that in vitro data of Omicron infection does provide neutralizing antibodies against Delta, for example. And Delta, if we look at the sequences, some people were worried there's going to be this rumbling Delta epidemic under the Omicron one, but in the sequence data, the Deltas have almost vanished. What's left right now is almost totally Omicron. And the other thing is that, Omicron's spike protein mutated massively. It had like 30 mutations, much more than the previous variants of concern we've all, come to know and worry about. And, that changed multiple things. I discussed it probably changed its entrance modality to cells. It totally changed whether antibodies bind to it, hence its immune evasion. But there are sort of physical constraints, I think, on how much you can mess with this protein and it will still be able to do its job. And so is it possible we'll break out yet again and have yet another variant? It's absolutely possible. On some time scale, it's even likely. But I am pretty hopeful that in the spring and summer in highly vaccinated countries, we might have a pretty calm period. And the question becomes, can we use that calm period profitably to be ready for what happens after that?
Thanks, Jeff. That feels like quite a gently optimistic note. I'm definitely going to take it as that. Nick.
Let me bring a pessimistic note. Seriously, without a global vaccination strategy, I do think we will inevitably get more variants. I don't see how that's not possible, if I'm honest. But I think Jeff makes a really good point. A lot of people will get a boost, irrespective of not being vaccinated, because they'll get an Omicron boost. I don't see how this will not end up a bit like we have with flu, where we just have to have an annual vaccination strategy. It will change. There will be variants. And that's why I think this kind of work is important, so we can fix up what our vaccination strategy should be for patients going forward. Because I don't think this is an 18-month problem. I think this is a 5-year, 10-year problem. And I don't mean that in a pessimistic way. We learn and we can adapt. And, if we get our vaccination strategies correct, we learn and we do well for our patients and everyone.
So what does this mean, Tariq, for our patients, our IBD patients? What message do we give them now? What's the future? What does it hold, do you think?
I think we have reason to feel optimistic based on Jeff's comments, which I agree with. I think that we are going to need to be adequately set up to move quickly, to continue to vaccinate patients regularly. We desperately need a correlate of protection so we can identify those individuals at risk, and we still don't have this. So that would certainly be helpful. I think the other point that Nick's made is really important, that although in the developed world there's reason to be optimistic, I think the majority of the world are still have a great problem with vaccine access, and that has impact not only on their own countries but also on the rest of the world in terms of the evolution of new variants. So I think we need to think wider than just the UK.
Thank you. So I'm taking cautiously optimistic as the general verdict. James Lindsay.
Yeah, can I ask, probably Nick, obviously we focused very much on antibody responses here, and of course they're clearly important, and you've shown that. But that's only 50% of the argument. What about the impact of drugs on cell-mediated responses? I seem to remember some data suggesting they're not quite the same.
Yeah, that's right. So we have looked at T-cell responses in Clarity, and we have PBMCs from VIP as well, so we can directly answer that. The data we have from Clarity suggests that they are dissociated. So if you have a poor antibody response, it doesn't necessarily mean you have a poor T-cell response. And there's also some signs of encouragement in that we didn't see a great deficit in T-cell responses in the infliximab recipients. If anything, it looked slightly better, which is not that there are some rationale for why that might be. TNF, the cytokine, drives exhaustion in T cells. If you block that, you might get a more robust T cell response. So that's encouraging. And I think Tariq alluded to this as well. We don't know what the right correlate of protection is. Is it an antibody level? Is it a degree of T cell activation? Is it a combination of the two? Do they both decay the same? So there's lots and lots of unanswered questions here. But having a good T cell response, I see that as a really good thing for our IBD patients. And one last thing to add to that, whilst we see these diminished responses of antibody responses in anti-TNF recipients, most patients do have an antibody response. They probably do have some protection. So it's not all doom and gloom in these patients, and we're getting good vaccination into these patients. And you can see the boosted response that they get. So it really isn't all doom and gloom, even if you're on infliximab and thiopurine, I would say.
Elsa Hart.
Thanks all. Question for Tariq. And it comes back to what we've been talking about in this meeting quite a lot. So long COVID in the IBD group. So obviously in the first wave, probably a lot were shielding and didn't get it, but a lot of them are going to be getting it. They have had it or they're getting it now in the Omicron. So with all the pathways we're thinking about with inflammation, fatigue and some of these features, any sense, I know you're working a little bit on that, any signals yet or not?
So, Elsa, we have asked participants of the Clarity study to complete a long COVID questionnaire currently at one time interval. We may go back to the cohort before the study closes to ask them to complete it again. So we will have data on this and we'll be able to correlate it with other outcomes. So that is a study that we need to do. We are waiting on complete vaccine data before we can start that, but I suspect in the next month or so we'll be able to answer that question.
Thanks.
We're coming to 7 o'clock. Nick, Jeff, Tariq, thank you very much indeed. That was really just wonderful. I mean, to me, this is, as Nick said earlier, this has been the best of UK research and clinical and truly actionable, meaningful data on record scale. So thank you for that. So I think that really just leaves me to close up now. Here we are at 7 o'clock. We've had a long day. I think it's been a really, really good day. So I want just to say a few brief thank yous. I want to thank you again to Tim Raine and Elsa Hart for co-creating this meeting. I want to thank all of the faculty who have joined us either in person here in the studio today or virtually, for being not just present, but being very present and enthusiastic and really supporting the meeting, the concept, and the themes that we've really been trying to deliver. I want to thank you, our audience, for being so engaged throughout the day. The audience retention numbers that we've seen on the online platform have been truly amazing. You know, over 800 people pre-registered, a significant proportion, at many hundred, present throughout the day. Thank you very much indeed. There will be on-demand content available on the platform from next week, and maybe a few teasers shared before then. There will be a certificate of achievement, or certificate attendance rather, achievement, whichever way you want to look at it, that you can download now from the site or claim via email. I want to thank Galapagos for the sponsorship for this meeting. We have been able to put this on with zero interference, which has been hugely refreshing from us. We have been able to create from scratch a meeting that I think has really hit a chord with so many people. And I want to finally thank Through the Eye for doing a really tremendous job throughout a long day and beforehand, and indeed afterwards in terms of the production. So thank you very much for your attention. This, we very much hope, will be an annual event next year. I look forward to welcoming many of you in person to Edinburgh for what will then be IBD Edinburgh 2023. For now, good night. Thank you very much. Goodbye.